Today, I'm presenting a recent study about partner specificity in ectomychorrhizal symbiosis, which our teams have been prepared for 10 plus years. So I'm giving this talk as a research professor in Mycology and Microbiology Center, Tartu University, and also jointly professor of biology in King Saud University. So later, when you have questions, you can contact me via email. The study I'm presenting is quite unique in this sense that it's the first exercise how to prepare, write and analyze an open study, and this is for education purposes. So it means that after we received the molecular data, I decided to do all analysis and manuscript writing publicly. So I kept a blog where I added results of different statistical analysis and also pieces of the manuscript in preparation. So it's almost in real time. And later also I added here the revision with comments by the referees and also the full rebuttal letter. And below is the web address where you can still find this document. And for a few extra days, it's also open for commenting of the revised manuscript. So for this study, we had 4 basic hypotheses in the first place. So first of all, different taxa of plants differ in the richness of ectomychorrhizal fungi. Later, we extended this also to other fungi. Second, relative specificity in plant-fungal interactions depends on both plant and fungal partners. Third, partner specificity increases with stand age and relative dominance of tree species, hinting that specific host trees tend to accumulate specialist fungi when these trees become older and more dominating the community. Finally, specialist fungi are mostly rare members of the fungal community. So this is mainly expectation based on previous studies. We had also 3 additional objectives, including finding reasonable proxies to quantify partner specificity. We also thought whether it's reasonable to consider plants and mycorrhizal fungi as equal partners. So this had quite positive impact from the journal editorial. And also we tried to distinguish the avoidance of preference and to distinguish preference and avoidance aspects in the overall specificity concept. So as a main background, we know that 3 species or 3 genera are very important regulators of communities of ectomychorrhizal fungi, also sometimes arbuscular mycorrhizal fungi, and sometimes also leaf endophytes and pathogens. But those previous studies have shown that there is very much that remains to be explained in those very different patterns. So in those figures on the top, you can see two studies, quite old studies, where the authors found that around between 10 and 15% of ectomycorrhizal fungi could be regarded as partner specific, and those specific analyses were actually redone by myself using the most appropriate statistical methods. There were certain outlying studies, as you can see in the right bottom, where in case of ulnus, when it's included in the mixed forest, then these forests show very strong patterns of specificity, where sometimes 90% or even more of the species can be regarded as specific. And all those studies have found, except for the last one, that the generalist fungi strongly dominate ectomycorrhizal fungal communities. Our own meta-analysis study also found that ectomycorrhizal fungi are comparably specific to plants, as so are other fungal guilds. And to some extent, we also wanted to revisit this claim based on this Northern Baltic white plant community study. So this new study actually partly relies on our previous study, which was published 3 years ago, where we focused on richness and compositional differences in fungal communities. So in that study, we sampled all terrestrial habitat types, more than 1,200 plots, with a standard design of sampling and also standard design of full-length ITS sequencing. And this study also addressed the basic questions in ecology and conservation of fungi. We selected long-read backbio sequencing for identification because using this approach, we could get the full-length ITS sequence. And the benefit of full-length sequence is more precise identification at the level of species. And nowadays, backbio also provides super accurate reads. It can be much more, it can have much less errors compared to Sanger sequencing and also Illumina sequencing. This approach also provides us less unknown taxa at the level of phylum or kingdom, and hence also more secure functional assignment of taxa to different fungal functional guilds. Based on this study, we also prepared a regional map of fungal richness. In blue colors, you can see relatively rich fungal communities, mainly located in mixed forests and neutral pH wetlands, whereas the poorest richness communities are located mainly in poor pine forests and especially in acidic bogs. We also addressed composition of ectomychorrhizal fungi and found that the main factor determining fungal composition is actually the host genus, which in a way also partly motivated this study. Also, soil pH and soil CN ratio had very strong effects on composition, and that's why we also included these as direct and indirect effects in the current study. So here we used also total soil approach by focusing on monocultures and different monocultures formed by different plant species. And in some occasions, we also compared these to mixed forests. So in a way, these studies are opposite to previous mixed forest studies, where mostly fungi were directly sequenced from root tips or isolated from leaves and then sequenced, and then the effect of plant was compared. So in our case, we cannot really say that there is it's like the fully realized host specificity. We can rather say that it is potential specificity, because from our approach here, we are in a way ignoring when the situations where plant roots are intermingling in soils. And for this particular study, we collected additional 200 plus plots in 2020 by mostly focusing on monocultures in different parts of Estonia. So altogether, we had more than 1,400 plots with ectomychorrhizal vegetation from Estonia and northern Latvia, and these included almost 500 ectomychorrhizal monocultures with a total of 39 woody plant species as the monoculture species. When it comes to fungi, we identified nearly 65,000 fungal species, out of which about 1/4 were ectomychorrhizal, and these numbers are related to 98% sequence similarity threshold. By trying several indexes for specificity, we found that the phi index might suit the best for the proxy of specificity. The benefits of this index is that it is ranging from -1 to 0 to +1, and unfortunately it is sensitive to frequency, so that this has to be taken into account in some occasions. We also took the R index of indication as an alternative proxy for specificity, because this is frequency insensitive, but it ranges from 0 to indefinite, and so it has not so good statistical properties. First of all, we tried to see what factors affect the fungal richness strongest. In the top figure, you can see the ectomychorrhizal fungi, and in the bottom figure, you can see the structural equation model on non-ectomychorrhizal fungi. So in both cases, plant richness has a strong direct effect on richness, but for non-ectomycorrhizal fungi, also ectomycorrhizal plant proportion has a negative effect. And also when we included plant genus as a categorical predictor, it has the second strong effect on richness on both ectomycorrhizal fungi and to a bit less extent on non-mycorrhizal fungi. And also plant richness effect was 5 times stronger on ectomycorrhizal fungi compared to non-ectomycorrhizal fungi. And interestingly, there was also an additional effect of fungal evenness, which had positive effect on richness, but many of the other predictors mainly affected evenness directly rather than richness. But this was not the case in non-ectomycorrhizal fungi. So the main effects were related to vegetation. The effects, all effects prevailed over stronger in ectomycorrhizal fungi compared with non-mycorrhizal fungi, and the effects tended to be stronger for richness compared to composition. There was a very strong joint effect of plant genus and plant phylogeny that was quite difficult to disentangle, but other analysis showed that the genus effect was really strong. So we consider that, especially in ectomycorrhizal fungi, the additional effect of phylogeny added just a little. But when it comes to composition, then the effect of phylogeny alone tended to be much stronger compared with the genus effect. So this is here one of the main figures before its tilted presentation. The figure shows that you can see the strongest or the highest richness of ectomycorrhizal fungi in birches, followed by oak and spruce monocultures. The lowest diversity we can see in certain Salix species, Pinus muga, Helianthemum, and certain Populus species. In red, there are shown introduced or non-native plant species, but when we compared the averages statistically, there was no significant differences. Here we also compare the genus level effects, again showing that birches have highest richness, and larch and alders have lowest richness quite distinctly. And in the left bottom, we have also compared richness of mixed forests. So the when there are more ectomychorrhizal tree species, the richness of fungi steadily increases. And we can roughly say that the birch, as the most species-rich monoculture, roughly equals to 4 species mixture in terms of fungal richness. But forests with more plant species, they do better than birch in that respect. Since the tree genus had the strongest effect on fungi, we continued mostly with this level analysis. And here we also show how tree genus affects richness of non-ectomycorrhizal fungi. So you can see that very differently from ectomycorrhizal fungi, the non-mycorrhizal fungal richness peaks in almost forests, and is also quite remarkable in large forests. And when we compare across all fungi, these contrasting differences between ectomycorrhizal and non-ectomycorrhizal fungi are evened out. So there are not very strong differences, but conifers stand out as genera with relatively low richness compared to deciduous trees. And when it comes to relative abundance of ectomycorrhizal fungi in terms of proportion of reeds compared with all reeds, again, larch and alder have relatively lower proportions of reeds, which may or may not be related to their relatively low richness. When it comes to specificity, which was the other major objective, we also built those structural equation models for ectomycorrhizal specificity at the level of stands. So here we have weighted specificity per stand and unweighted measures per stand. So these operate quite similarly, but mostly those weighted measures give stronger models and stronger results. So the main predictor of fungal specificity was differences in plant genus, and other factors had relatively lower roles, especially the fungal evenness, which was mainly affected by fungal richness and the plant richness. And this figure actually shows that ectomycorrhizal plant richness actually indirectly affects fungal specificity through the richness and evenness of ectomycorrhizal fungi. So when we try to put ectomycorrhizal fungi in the context of all fungi, we can see that the preference side in ectomycorrhizal fungi is relatively high. It's also surprisingly high in root endophytes, and it's relatively low in avascular mycorrhizal fungi, but also in non-plant parasites and in pollen saprotrophs. So in almost all these cases, except pollen saprotrophs, preference is stronger than avoidance. So preference here means that there is a specific group of plants that fungi are linked with, And conversely, avoidance is a situation when there is a certain plant group that is discriminated against or avoided. And also in avoidance, it was relatively uncommon when avoidance was stronger than preference. There were only 4 cases in all ectomychorrhizal fungi that were statistically tested, but avoidance was quite strong in pollen saprotrophs and also in molds, quite surprisingly. And lowest avoidance as preference was observed in avascular mycorrhizal fungi. So when it comes to patterns of avoidance, we can also see that the strongest avoidance occurs in Tilia and also in Populus and Corylus stands. So this stands in contrast to preference, where the alders had the strongest preference patterns. And here we have another analysis across all ectomycorrhizal fungal species. And this analysis actually shows that across all species, Alnus tends to be the one that is most strongly avoided. And this is in line with previous studies showing that certain fungi strongly associate with Alnus, and there is another group of fungi, much more common group of fungi, that never associate with Alnus. And these figures here actually show that specialist fungi dominate both in terms of richness and abundance. So the relative abundance is indicated by columns. The average abundance of specialists is about 78% across all monocultures. In mixed forest, it is less. It's around 58%. And the richness is a bit less, indicated by those single lines. So this shows at higher host plant abundance, also fungal species that are specialists, they are relatively more abundant. And the bottom picture shows that Alnus accumulates host-specific fungi. So this figure is based on the average fungal specificity per particular plot, and these plots are separated here by specific monocultures. And then the monocultures have much higher average specificity or indicator values compared with mixed stands. For this revision, we also analyzed preference for specific plant partners for all major fungal guilds. And these analyses here show that principally all these fungal guilds differ in preference of certain plant genera. When ectomychorrhizal fungi tend to have highest specificity for ulnus, then there are many other groups that have specificity for pine, except and also for spruce in many cases. Other fungi, other plant genera have less specificity in fungi. This figure compares preference in plant species for ectomychorrhizal fungal lineages. So each column indicates a different tree species, and each color here indicates a different ectomychorrhizal fungal lineage. So, for example, pine and spruce, the two first columns, have relatively low richness of Tomentella thelephora, which is the dominant ectomychorrhizal lineage for both Ulnus glutinosa and Ulnus incana. And when the top figure shows the relative richness patterns, then the bottom figure shows relative abundance patterns. And you can see that in those extreme cases for conifers and also for Ulnus, patterns based on abundance are stronger. This supplementary figure indicates relative partner preferences in ectomychorrhizal fungal lineages, which are shown as rows, and the preferences for different groups are weighted by the relative abundance of these monocultures or mixed cultures. So here the yellow bars indicate mixed cultures, but it's quite interesting that only one of the lineages, Serendipita 2, is relatively more common in mixed cultures compared to any monoculture, whereas many groups such as Amphinema, Telospora, Polytopsis, Hydnellum sarcodon, they prefer strongly one or the other ectomychorrhizal plant genus or plant species. The similar figure based on fungal genera, which are here also listed as rows. And again, this figure shows that fungal genera differ in the proportion of specialists. Here, the genus Alnicola has the strongest preference across all fungal genera, and most of its species, as indicated in red, are specialists of Ulnus. In the case of Amphinema and Telospora, the main host is actually Picea, and in the genus Sullus, the only preferred host is pine. It could be also Pseudotsuga or Larix, but these two hosts were excluded from this analysis because we did not have enough data to analyze this statistically. Here again, we have compared the relative indicator value of species belonging to different fungal genera. And here again, the genus Alnicola and Tylospora prevailed. Quite interestingly, also Bacillus shows relatively high indicator values, but this is related to the fact that both the alder-associated Bacillus filamentosus and the common host genus species Bacillus involutus are actually divided into several cryptic species that have quite strong partner specificity. And here the blue and red circles also indicate the relative proportion of strong specialists and overall specialists. Strong specialists or strong indicators we define as taxa for which the indicator value had p-value support less than 0.001. In this figure, we are comparing different indicators. Strong indicators are indicated in red lines, weak indicators in orange lines, and generalists or indicators of none are indicated in black lines. So these figures show that the relative proportion of especially strong indicators is responding to relative abundance of the dominant ectomychorrhizal tree species, and this relationship becomes especially strong when the relative abundance of one tree species exceeding 90% or actually even 95%. This pattern we also see in the when we consider the relative abundance of these specific ectomychorrhizal fungi. And what is interesting is that the weak indicators with a p-value between 0.05 and 0.001, these virtually do not respond to the abundance of trees. And what is also interesting is that the strong indicators are mostly located in areas that have either very low or very high pH. This is difficult to explain why we see this pattern, but one explanation could be that certain host tree genera occur only in very acidic or relatively neutral soils, and this is partly also reflecting the plant effect. We also compared these patterns or relationships to soil pH and also vegetation age among different tree genera, and we can see that different tree genera, as indicated by different colors, differ in these patterns. This black symbols indicate pine, and you can see that actually most of the pine plots are located in the very low acidity, and they are also related to higher than average indicator values. Whereas those pines that are in alkaline soils, their average indicator values are very low. Similar analysis with soil age. Across all taxa, soil age did not have a reasonable effect on accumulation of indicators, but we see that in certain plant genera, for example, pine, oak, and purple one, I think it's Tilia. These are the fungal communities associated with these trees become more specific with increasing age. We also used this R index to address whether there is a relationship between host specificity of fungal species and their commonness, as this metric was independent of sample size or frequency of observation. These plots show that there is no biologically meaningful relationship between the fungal species frequency or the fungal species relative abundance and its indicator value. But we could show that especially the rare species, which were found in 4, 5, 6 occasions, they had much smaller pH range compared to more frequent species. So notice the two base logarithm scale in this figure. So you also must note that the species found 4 or 5 times here in most studies, which use typically 10 or even 100 times less samples. In most of these studies, these species would be so rare that they would not be found at all. So this is here also one complicated graph where again different colors indicate the relative indicator value of fungal species, with red being strong indicators and black ones no indicators. And blue are indicating the very rare species that were actually occurring only in 3 or less sites. So these could not be analyzed for their specificity values. So we plotted the accumulation of all these types of species by the richness of plants and also accumulating richness of fungi. So these graphs show that as plant richness grows up or fungal richness goes up, then all these groups also increase. But when we consider the relative abundances of these particular fungal groups, we actually see that the strong specialists decline when the total number of ectomychorrhizal fungi in these sites are growing, and by contrast, the number and proportion of generalists is growing. So it implies that it is mostly generalists that contribute to the most rich ectomycorrhizal fungal communities. And here are the main conclusions from this partner specificity study. So first, the specialist fungi are much more common, both in terms of richness and relative abundance than expected previously. But the so-called avoiders and exclusive specialists or super specialists are quite rare. Specificity in fungi is strongest at the level of plant genus, but we can see also increasing patterns of specificity from the level starting from subgenera to families and eventually class. We also see that specialists tend to accumulate with increasing proportions of their host plants, and this is not a linear process but an exponential process. Both in fungi and plants, the relative specificity is unrelated to their commonness or rarity. I didn't display the similar graph for plants. And as mentioned for the last slide, generalists rather than specialists contribute to hyperdiverse ectomychorrhizal fungal communities. So what we can conclude for conservation is that old mixed forests and mixed parks deserve most conservation from the perspective of ectomychorrhizal fungi, but also old monocultures such as old spruce or pine stands deserve conservations, and any stands that are dominated by rare tree species, such as forests dominated or even copies dominated by certain species of salix like Salix alba or Salix cinerea, Salix fragilis. These certainly deserve preservation because they may be the only places that harbor certain specific fungal taxa. So thank you for the attention, and I thank all my colleagues who contributed to this study, and also the founders, Silvanova, Estonian Science Foundation, and Norwegian Baltic Financial Mechanism.